When I first read through a microbiology textbook and I got to the chapter on bacterial growth and microbial growth curves, I thought, I've directly experienced this in my yogurt making. So that's what I want to do today is I want to make yogurt with you. And as we go through the process, we're going to talk about some classic fundamental microbiology concepts. Personally, I love yogurt. It's way more than just a trendy snack. In fact, historians think it's been around for over 5,000 years and probably happened on accident. People used to store milk in things like animal skins and clay pots, and bacteria from the environment would naturally just ferment it. So this was happening long before people even knew what microbes were. What most people don't know is that you can actually make yogurt from many different bacterial species. You can use pretty much anything that's happy eating lactose, which is the main sugar source found in milk. Today we're going to create yogurt from both a store-bought active culture and also from some probiotics. So this is going to let us play around with the idea that fermentation times are going to vary based off of whether our culture is active or dormant, and even amount of starting culture, so amount of viable cells that we're going to start with. So let me present to you our lineup. We have this store-bought yogurt, it's my favorite, and it also has two strains that are really cool. We have a Streptococcus thermophilus and Lactobacillus bulgaricus, and these two are really cool because they have a little bit of a symbiotic relationship going on. Next is a medical grade, Next is a multi-strain probiotic, and this one's pretty fun because we have, I believe, at least 8 different species and a really high starting CFU, which is colony forming units. So in one capsule, we should have about 112.5 billion bacteria. That's a lot. And then I have a single strain probiotic. This is Bacillus coagulans, and they even have the strain on here, which is great. This one has lower starting amount of bacteria, 2 billion CFUs, so big difference. And another notable thing is that this one is shelf stable, whereas this one is refrigerated. The shelf stable one is likely in the form of endospores because Bacillus bacteria can form endospores, these really hardy resting structures. This one is refrigerated and probably freeze-dried. So we've got a couple of different variables we'll play around with. Like I mentioned, active culture versus dormant, and that's going to affect fermentation times. So real quick, I'm just going to out myself on something here. We're changing many variables at once, so this is not exactly sound science, but I don't know. We're in my kitchen and this is what we got, so we're going to roll with it. Like a good microbiologist, my first step is to disinfect my workspace and sanitize my equipment. The cool thing about an Instant Pot or other pressure cookers is that they're sort of like mini autoclaves. An autoclave is a piece of lab equipment that can sterilize materials by exposing them to high-pressure saturated steam, effectively killing microorganisms. I'm putting the jars I'll be using into the Instant Pot over a thin layer of water and using the steam function for just a few minutes. The Instant Pot will pressurize, allowing the water inside to reach a temperature closer to 240 degrees Fahrenheit, or about 115 degrees Celsius. And that's higher than the boiling point of water at sea level, which is 212 degrees Fahrenheit or 100 degrees Celsius. But being realistic, I don't expect anything to be sterile today. I'm working in my kitchen, not a laboratory. While I wait for the jars to cool off, I'm getting organized and labeling my jars. You can add milk actually directly to the Instant Pot, but here I'm using separate jars so that we can compare how each culture ferments. For now, we just add milk because the first step is to heat up the yogurt to at least 165 degrees Fahrenheit, or about 74 degrees Celsius. This will not only reduce unwanted microbes in the milk if they're present, it will also cause some of the milk proteins to denature, and this creates a thicker texture later on for our yogurt. I don't want to add my culture to the milk while it's still super hot, or it can kill my microbes. So I've got to let it cool off to about 106 degrees Fahrenheit or 41 degrees Celsius, which is the temperature that we're going to be incubating at. Now let's add some microbes to this party. On deck, we've got the fridge-stable multi-strain probiotic. I just pop open the capsule, pour the powder directly into the milk, and whisk it with the tiniest whisk you've ever seen in your life. You'll notice I'm also pouring boiling water over the whisk between jars, and this is just to try to reduce cross-contamination. Next up is the shelf-stable Bacillus coagulans. Same process here. And finally, I'm adding a tiny spoonful of my favorite store-bought yogurt. I don't know the concentration of microbes in this yogurt, but from experience, I think I'm adding a generous amount. All right, time to just let it do its thing and ferment, and we'll check on it in a couple of hours. We're about 2 hours into the fermentation process, so let's check on our jars. The first thing you'll notice is maybe a whole lot of nothing. It just looks like milk still. That's because our microbes are in what's called the lag phase of the classic microbial growth curve. In microbiology, it's common to use growth curves to describe microbes cultivated in a batch culture, meaning that they're incubated in a closed vessel, like a test tube, a petri dish, or in the case of my yogurt making, a glass jar in an Instant Pot. The key here is that there's no replenishment of nutrients or food sources for the microbes. Bacteria reproduce by an asexual process called binary fission. So growth, when it's rolling, is exponential and plotted on a logarithmic scale over time. The curve has distinct phases, with the lag phase at the start. You might be guessing that maybe the bacteria are inactive during this phase. Surprisingly, it's not inactivity. Instead, the cells might be doing a number of things. Maybe the cells are depleted of ATP, essential cofactors, or ribosomes, or essential cellular components that they need to make more of before their growth can really begin. Maybe the media is different. When we move microbes from one media to another, new enzymes are needed for a new nutritional base. For example, if the bacteria has to switch from glucose to lactose as a sugar source. Without the right enzymes, no microbe can metabolize or eat the lactose in milk. Or maybe the bacteria were stressed and need time to recover and repair. This is likely the case for our capsule-based probiotics. The Bacillus coagulans species in particular is an endospore-forming bacteria. This probiotic capsule is likely chock full of endospores, and those spores need to germinate into what we call vegetative cells before they can start growing and multiplying in number. So we're at the 4-hour mark, and I want to check in on this yogurt and see if anything has started to develop. Okay, the first one is basically yogurt. You can see that when I tilt it, it's really slow. It's definitely coagulating, and that is surprising. So this jar is our multi-strain probiotic. Plot twist, okay? Number 2, the Bacillus. We've got pure liquid. I'm not surprised. I think this one might have to ferment for a long time. And then number 3 from the yogurt. It's getting there. It's still liquidy. I wouldn't call this ready by any means, but it's starting to solidify. Okay, so since we're in my kitchen, this is really the only measurement that I'm going to be using to see if these bacteria seem to be moving into the next phase of our microbial growth curve, the log phase. During the log phase, growth has really picked up. The growth rate is constant, meaning that the bacteria are completing the cell cycle and doubling in number at regular intervals. I'm saying doubling in number because bacteria grow by asexual binary fission. It goes a little something like this. So the bacterial cell typically has one circular chromosome. Each chromosome has a single site at which replication starts. This is called the origin of replication, or sometimes just simply origin. The terminus is where replication is completed. So the origin and terminus move to the middle of the cell, and proteins needed for chromosome replication also assemble at the origin. The DNA synthesizing machinery is called the replisome, and DNA replication will proceed in both directions from the origin. As the chromosome is replicated, the two origins move towards opposite ends of the cell, and the rest of each chromosome follows. Septation is the process of forming a cross-wall between the newly formed daughter cells. Here's a rough sketch of the steps. So first we have a selection of the site where the septum will form, and then there's the assembly of the Z-ring. The Z-ring is a polymer of the cytoskeleton protein FTSZ. That's where it gets its name. There's actually also this whole system called the Min system, which oscillates between the poles of the cell like a herding dog, and it keeps the Z-ring aligned and deters it from polymerizing at the poles of the cell. Then we've got the assembly of machinery for synthesis of peptidoglycan, which we know is the cell wall main component, and other cell wall constituents. Finally, there's the constriction of the cell and septum formation. And there you have it. We go from 1 cell to 2. Because the growth rate is constant and bacteria divide by binary fission, we see exponential growth when we're in the log phase. Again, that's why we're plotting these growth curves on a logarithmic scale. 1 cell becomes 2, 2 becomes 4, 4 becomes 8, and so on. So I thought you might be wondering, how do I know that we are in the log phase and fermentation is happening just from the fact that the milk is a little clumpy? Well, when I say fermentation, I basically mean that these bacteria are eating up sugars in the milk. And they're doing that for the same reasons that we eat food, right? To obtain energy and eventually reproduce. So as they're fermenting, they're eating these sugars. They're also creating byproducts like acids. Lactobacillus, for example, creates lactic acid when it ferments. So as these acids start to accumulate in the milk, it causes proteins found in the milk to denature and essentially clump together. And that's why we get a big change in texture. So during the log phase, we are seeing a metabolic party, essentially. ATP is being used and made. We are seeing the accumulation of acid byproducts and changes to texture and flavor. And that's fermentation in action. Here's something cool. We can actually calculate an estimate for how many bacterial CFUs are in our yogurt jars at a given time if we know the starting number of cells. Let's use our microbe Bacillus coagulans, strain GBI 306086, as an example. Also note that the quantitative aspects of growth are only valid for cultures in the exponential phase and only apply to microbes that divide via binary fission. So we can take a simple equation. N sub t is the population at time t. N sub 0 is the initial population number. For this microbe, we know it's 2 billion CFUs. And then lowercase n is the number of generations in a given time t. Let's say we want to know how many microbes, or more accurately, CFUs, are in the yogurt jar after 4 hours of fermentation in the log phase. Time would be 4 hours, and the generation time, which is basically the amount of time it takes for Bacillus coagulans, a single cell, to do binary fission and become 2 cells, is about 20 to 40 minutes, so let's call it 30 minutes. So 30 minutes goes into 4 hours 8 times, so that gives us our n. So we take 2 billion and multiply that by 2 to the power of 8. And that gives us a really massive number. So in just 4 hours, there would be 512 billion CFUs. What would it be like for our yogurt here if we did let these microbes progress on their fermentation journey in my Instant Pot? Well, the next stop would be the stationary phase. At this point, overall bacterial count plateaus. For bacteria in the lab, a general population number for when bacteria hit the stationary phase is about 10 to the 9th cells per milliliter. That's actually a really massive number, so just let that sink in. But here's the puzzle. There are bacteria in the culture during the stationary phase that are still actively growing. So why is the number plateaued? Well, the answer is that there are cells now reaching the end of their life cycle and dying at the same time that new cells are being created. Beyond the stationary phase, we dramatically enter the death phase. You can guess what's happening to our little microbes here, but why? Maybe the most obvious reason is nutrient limitation, which already started to become a problem in the stationary phase. In a batch culture in lab, or like in my yogurt here, I'm growing microbes in a glass jar filled with a finite amount of nutrients. Eventually, suitable food just runs out for these guys. Maybe less obvious is that there's a buildup of toxic waste. I think this part's actually pretty cool. During fermentation, these microbes are, to simplify, eating up lactose, and as they do, they create byproducts like lactic acid. So, as fermentation progresses, the pH drops. The buildup of toxic waste and acids causes harm to cells in the culture. Pair that with nutrient deprivation, and the number of viable cells declines exponentially, with cells dying at a constant rate. So the yogurt at this point, if I can even call it that, would be pretty gnarly. And just to give it mention, the long-term stationary phase is a really interesting phenomenon. The harsh conditions of the death phase puts a selective pressure on that dying population. Throughout that stationary phase, we see successive waves of genetic variants able to weather the storm, though I wouldn't say that they're thriving. So, what were the results? How'd the process go? Well, not surprisingly, there was some variation between our tests. And one reason for that is that there's various ways to control microbial growth. What I mean is that the manufacturers of all of these products are trying to create a product where the microbes are alive but metabolically stagnant. So, for example, even in the yogurt where we have active cultures, we refrigerate it so that the microbes are slowed down to a point that we're not changing the texture and flavor of the product. You can compare that to this one, which needs to be metabolically slowed down enough that it's actually shelf stable. So because the Bacillus strain is shelf stable, I'm guessing that it's in the form of an endospore, which Bacillus coagulans is known to form naturally when faced with environmental stress. So it makes a lot of sense that this would have the longest lag phase and kind of take the longest to wake up. I actually let the Bacillus yogurt ferment overnight because by the time that I took these two out of the Instant Pot, this was still milky. So I don't know exactly when we started to see signs of exponential growth, but by the morning, about 17 hours of fermentation, and we've got yogurt, though after tasting it, I see why it's not a common yogurt strain. The microbes for our multi-strain refrigerated probiotic were probably lyophilized, which is a fancy word for freeze drying. So in this process, the microbes are rapidly frozen and water is removed via sublimation under vacuum. So we end up with microbes that are alive but metabolically inactive, mainly because they lack water, which is the basis for chemical reactions in cells. And then our yogurt, on the other hand, contains active cultures, which significantly reduces lag time and can maybe eliminate it in some cases. So that is why the biggest plot twist for me was that our fastest to reach exponential growth was the probiotic. And I'm gonna chalk that up to the fact that this had the highest starting count of CFUs. It was at 112 billion. Not super sure for this one, so can't do a fair comparison, but that's quite high. Just to put that in context, we can use the same math that we did before to see how long it would take that Bacillus culture to reach the starting CFU count of the other probiotic, at least once it makes it to the log phase. So we set up our equation just like we did before, but this time we're solving for n. After some simplifying, we get 56.25 equals 2 to the power of n, which means we've got to pull out some logarithms. So solving for n gives us about 5.814. Now, that's the number of generations, not time. We said that generation time for Bacillus coagulans is probably somewhere around 30 minutes. So accounting for that, it would actually take almost 3 hours of fermentation in the log phase for that Bacillus yogurt to reach the starting concentration of the multi-strain probiotic we used. And then remember that growth in the log phase is exponential. So yeah, the multi-strain probiotic really had a leg up in this process. So there you have it, foundational microbiology concepts, growth curves, growth kinetics, all applied to yogurt making. And special thanks to my dog Boa for not barking during the filming of this video. 11 out of 10.